Okay, folks, so we're on to the next one. This is an interesting one. So it's talking about leveraging plant-driven insights for smarter cultivation and yields. Our current king, King Charles, talked about talking to plants. Here we're thinking about maybe listening to the plant signals, but actually using it for more sensible applications. So we've got Julien de Giorgi. He's at Vivent, I'm not quite sure, depends where you are, how you pronounce this. Julien, it's all yours. Looking forward to hearing this.
Thank you. Good morning, everyone. So my goal today is not only to introduce you the company, Vivent Biosignals. I want also to introduce you to our technology, our sensor, but I think more importantly, I want to show you the applications we can with such technology in the context of CEA and indoor farming. So a few words about the company. We are based in Gland in Switzerland, but we have also offices in Zeiss in Netherlands. We are a mix of plant data scientists, agronomists, software engineers, but I think we are all working around the same idea, making plant data accessible and actionable for growers. To understand a bit what we do, let me just first show you where we sit in the bigger picture. CEA has made incredible progress over the last 200 years. Each major advance, better structure, better nutrient, better light optimization, digital optimization, all of that result in a significant yield improvement with a higher production. I think it was mentioned in the first presentation that all this discovery after a few years, all of them are reaching like a plateau, which means that we cannot produce anymore. And at that stage, usually we are a bit forced to innovate, to find like a new qualitative jump in the process. And now we are entering, I think, and that's what we believe in Vivent, we believe that the next qualitative jump is what we call plant-driven agronomy. And this is about capturing and understanding the data coming from the plant itself. And this is exactly where Vivent Biosignal is operating. So at Vivent, we think that, I think we will all be agreed that the plant is, I think, the best sensor. As you know, plant cannot run away from a stressful situation. They evolved developing a whole range of receptors to perceive the environment, and they also developed at the same time a range of internal communication system to propagate this information. One of these internal communication, these internal signals, are what we call bioelectrical signals that we name also biosignals. So those biosignals, they are charged particles that are transported through plant tissues in response to any kind of stress or change in the environment. So the video that I'm going to play is showing exactly that. You can see a plant being attacked by an insect, and you will see a calcium wave spreading from the attacked zone towards the other leaves. This is not just a reaction. This is a signal sent to the other leaves to trigger a defense mechanism. And this is, I think, a very good example of what we call a biosignal. On this video, probably the most interesting part is the time range. Here you see that this information is propagated into the plants under a minute. So now the question, so at Vivent, if I have to describe Vivent, we are experts in extracting this data, measuring it, and transforming that data into agronomic insights. So Now, next question is, how do we measure these biosignals? Maybe I will invite you to just quickly look at my camera. This is our sensor. It has a name. We call it Vita. It you can see it as a ECG for humans. So it's connected to a set of electrodes. Those electrodes, they will be connected directly in the plants. And once I press the button, it starts recording immediately those electrical signals. Those signals, they are sent to our cloud. On the cloud, they are translated, they are decoded by machine learning and algorithm, and they are transformed into agronomical insights. And those agronomical insights can be visualized on our dashboards. So a few words about our dashboards. So growers have access to our machine learning model directly on screen. We have like a real-time representation, as you can see here. So they can have an idea of the plant's physiological status as any time in real time. And it also displays historical data that can be visualized over different time windows. So here it's like a week. And it allows growers to not only see what is happening in real time, but also to look at the trend. The dashboard also gives the possibility to compare different sectors, to compare different greenhouse treatment varieties or conditions, which is quite useful when running trials, for instance, or managing multiple growing environments. In this example, so in this dashboard example, you see what we call one of the machine learning we are using that we call water status. It's an indication of how much the plants needs water. So if you go in the green, it means that the plant doesn't need water, and if you go in the red, it needs water. But we have all sorts of different machine learning model that can predict and give an information in real time about the status of the plant. So we can look at the drought stress, we can look at nutrient deficit, but also pathogens. So I want to give you 4 agronomical applications of this technology. And the first one is what we call plant-driven irrigation. So the question we asked was quite simple. Can plant biosignals, can those biosignals be used to control irrigation? Like another way to ask the question is, what is happening if we let the plants decide when to irrigate? And how is it performing compared to, I would call it like a conventional irrigation strategy? So we decided to test that. So we take like a set of plants in a greenhouse. We let the growers decide on the irrigation strategy. So we look at the environment around the plants, like the balance, the EC, all the parameters, and it just go with the irrigation. And in the same greenhouse, we connect the plants with the sensor. We look at the water status that I just showed you before, and we automatize the system to let the plants trigger the irrigation during the day and sometime during the night. And so we performed this type of trial in a cucumber greenhouse in collaboration with Zone College, and also in tomato greenhouse in collaboration with Tomato World. And what I'm going to show you now is the results. Let's start with the cucumber. We looked at the water used and the yield. So in terms of water used, you notice that we use 20% less water when we let the plants decide of the irrigation. You see that this number of cycles per day was, generally speaking, lower compared to the conventional irrigation strategy. But most interestingly is what we see with the yield. We get 12% more pieces. The pieces were from of the same quality. So we ended up with, in total, 120 kilos with the, if we let the plants decide, but with the conventional, we got like 100 kilos. What I want to say here is that these results have been reproduced this season. At the yield level, it's very similar results. And now I'm going to show you the tomato results. So with tomato, I cannot share the exact numbers yet. We are currently reproducing this trial. So we just want to confirm first what we observed on the first one, because what we observed on the first one is that, interestingly, we use slightly more water, but we ended up with a higher total yield, kilos per meter square, bigger fruit also. Fruit weight was bigger, but also we have lower fruit abortion percentage. But I would say in tomato and cucumber, I would say that the conclusion is very similar, is that if we let the plants decide on the irrigation, it gives more fruits. It's more generative. Something I want to say here that I think this is quite important to mention. The conclusion here is not to say, okay, we are doing better. Here the conclusion is, if we give the growers an additional information, this biosignal information coming from the plants, it can significantly improve and optimize irrigation, but also improve performance of the plants and production. I'm moving to a different type of topic now. We are still in a greenhouse in pepper, and here I'm looking at the daily plant activity. This daily plant activity, again, is measured with the sensor, and we are looking at the biosignals, and we use an algorithm to, to, to display the activity of the plants. And what you see here, in collaboration with Solom, in a greenhouse in Leamington in Canada, we compare different light spectrum. So we get one group with more red, we get one group with more blue, and you see with the sensor we can see which one of them are performing the best, which one are the higher activity. Here we change the spectrum for the red group. You see that it's catch up a bit. So at that stage, the two groups were at the same level of activity. And what is interesting is that when we switch the spectra, we have opposite results. So a consistent result, meaning that the blue spectrum still have more activity. So for growers, this means that they are able to optimize their lighting strategy based on how the plant actually responds. So adjusting spectrum, intensity, photoperiod, and seeing the plant's physiological response to each change. To stay in a lighting strategy. Here again, in similar greenhouse, here we are in tomato. The grower decided to compare two lighting strategy. So we got like what we call like a new one and an old one. And you see, during the comparison, we see that the old strategy was performing less compared to the new one. So just a few words about this metric, because this is quite an interesting one. The plant activity index is a metric to measure how well the plants is maintaining its physiological balance under its current conditions. So if it's in the green, it means that the physiological balance of the plant is very stable, and in the red, it means that there is a lot of stress. You can think of it as a real-time comfort indicator for the crop. And you see that by comparing these two strategies, we see that one strategy has a more optimal hours, which means that the balance of the plants was more stable, so it was spending less energy into adaptation. So, and after that, this new lighting strategy was also associated. This is maybe like the most interesting in this data. This low performance that we see here was associated with a significant lower occurrence, a significant higher occurrence of oedema. So after that, the growers decided to switch all the sector of the greenhouse with this new lighting strategy. So this is an interesting metric. So you can see when your plants are being stressed. Here in in the context of LED management, but we can also look at other parameters. Here I'm showing you a greenhouse also. And you see with the same index, we see immediately when the plant is reacting to something. Here, it's mean that the plant is being stressed, highly stressed. And when we compare this, when we compare that with the growers, we notice that the heating temperature was increasing too much in the morning. And after few adjustments, you see that the growers managed to keep the plants in a more optimal balance. Now I'm showing you another machine learning model that we developed that is related to nutrients. So we can see when the plant needs water, but we can also see when the plants are lacking some nutrients. Here I'm showing you a model that we call calcium status, which will indicate when the plant is entering in a deficit state. And you see that in this greenhouse in tomato, we see that the models start to indicate a downward trend on March. Here. But visual symptoms are only observed around 20 days later. So 20 days during which the growers could adjust calcium, could make something to improve that. So I think what is the take-home message of this slide is that we can detect potential deficit of your plants 20 days before you will see it on the plants. And what was interesting here is that it was missed by all other parameters. We look at the EC, we look at the concentration in the leaves, everything was okay. The only sensor that were detecting that was the Vivent Biosensor. So it gives really like a big window for the growers to act before observing those problems. I give you another machine learning model. In this case, this model is trained to recognize iron deficit. And you see, this is an interesting case because just after installation, we observed that at the growers, so this is strawberry grower, one of the greenhouse, one of the sector was immediately flagging iron deficit. And the growers adjust iron in the fertigation around that time. And you see that within 2 days, we see that on the models, there was a full recovery. And again, sensor was detecting this deficit here, and it was also observed by the growers. The plants was a bit more pale, and he decided to not wait to send the leaves for subflow analysis. He decided to immediately act and change the fertigation strategy. So I would say, okay, I talk about detection of nutrient deficit. We can optimize irrigation. We can look at plant activity. And one other aspect, last aspect, is detection of pests and disease. Those type of models are still in development. You easily understand that it takes more time to be validated. But in collaboration with Wageningen University, we perform a trial to see if we can detect thrips infestation on plants. And this is the result of the models. We were able to detect with a 72% accuracy which plants were infested by thrips. And you see again, like the time windows is very short. Like this is the infestation date, and we can observe that after 3 days, after 2 or 3 days. So here I'm showing you another one. This is for mildew. So again, shortly after inoculation, before any visible symptoms appear, the biosignals model start to picking up the plant's response. You see, and here you can detect like the infected group again, like few days after infection. So we have worked with a different set of pests, like nematode, corn root worm, sting bugs. it takes a bit more time to be developed, but you can really see like the potential of this technology for early detection of pests in your crops. So if you are thinking at the main problem in your greenhouse, I have no doubt that in these biosignals, we will find this signature and we can potentially develop a model for this specific pest. Okay, so how growers are using this technology? Growers integrate this system at the very different levels. Some use it as a daily monitoring tool, checking the dashboard every morning as part of their routine. Other, they use this to optimize their irrigation strategy, to evaluate how the crops respond to change in their LED recipe, or to validate whether climate adjustment is having the expected effect on the plants.
Mm-hmm.
So what is encouraging with all these stories of growers is that the more they use it, the more ways they find to integrate that into their agronomic decision. So to summarize, one sensor continuously reading what the crop is experiencing. Using plant biosignal, we can support irrigation decision, evaluate how crops respond to climate and light conditions, detect nutrient imbalances before visual symptoms, this is important, and in the very near future, I think provide very early warning for biological stressor. So yeah, if you are a grower, a researcher, like a company working in CEA or indoor farming, yeah, feel free to scan the QR code for a website and LinkedIn, and yeah, I will stay available on the platform if you have question, and
okay.
Yeah, happy to take the first question.
Thanks, Julien. We have a lot of questions, actually. There'll be more questions than we have time for, but one person's put the same question in multiple times. Asking, do you have sensors for all the nutrients? Is that how this is working? Or is it calibration curve based on specific nutrient use and you're just using your sensor to look at the cascade?
Yeah, so this is yeah. Here I need to enter a bit more about how we develop those this machine learning model. Let me maybe use this slide. So whatever type of stress, if we want to develop a model, we have to set up like a trial. So let's talk about the calcium model that I showed you before. Before being implemented on the grower dashboard, we have a set of different trials to train this model. So what we do is that we take a set of control plants that you have here, and we take a set of we call stress plants. So in this case, we will just decrease the calcium until we see deficit. So we will measure like the leaf tissue analysis. We will measure also the drain, the EC, and we will tell the machine, okay, look at the biosignals in these two set of plants, and we ask the machine to find the specific signature linked to this deficit. And then this is where the machine learning is important, because it will start training the model. And after training a first version, we have to test it. So we will use another set of plants that the model never see.
Yeah.
And we will see how efficient is it working. And if after validation, when we are very comfortable and highly confident about the models, we will implement it on the dashboard.
Cool. Okay, so an obvious one that goes back to money then is how many sensors do you need in a kind of, what's the per meter square equivalent you're going to need? You're not going to link every plant up, obviously. Mm-hmm. What have you got an average feel for this?
Yeah, so of course we are taking into consideration like the biological variability, right? So we don't want to take any decision based on one sensor, because it's like if you enter in the greenhouse, you see one plant and you decide to change your strategy based on that. So I would say the minimum amount of sensor for one greenhouse, 1 hectare, I would say 8 plants.
Okay.
8 plants that we will place either in a very representative zone of the greenhouse, or we can spread a bit the sensor all around the greenhouse. Some other people are more interested they have very specific issue with a certain greenhouse, so they will decide to put the sensor at a very specific place to get insight from this area. Yeah. But if you want to monitor like like the greenhouse in general, I would say 8 sensor minimum.
Okay, right. There's multiple questions left. Can I urge you, if you have more questions, to contact Julien in the backstage, the networking bit. And in fact, networking's opening up now. Thanks, Julien, for this. Generated huge number of questions. So I suspect people will be questioning you, which is good. We look forward to hear developments of this again. Next session will start at 11 o'clock, but please go back to the networking piece, contact Julien and the other speakers, and we'll catch you at 11. Thanks. Thank you, Julien.
Thank you. Thank you.